Hello, I'm Nate Russell. I'm a senior in operations research. The work I've been doing for since the start of this last summer and proceeding into now has been in high throughput and as well as next generation sequencing techniques for bio applications. So the general problem is that, well, we can now collect a lot of data at previously unthinkable scales of metabolic data as well as maybe GWAS studies. So you can collect all of this data. You have many features to describe a single patient or a single animal or a single molecule. But how do we analyze this in a statistical way when you have far more features than you have samples? Because although we can sequence a genome, it can cost many thousands of dollars, and so you're restricted in the sample sizes that you're allowed to acquire. So this creates a quite difficult p greater than n machine learning problem of discovering what features in a data set. To make this more real, what bacteria in your gut is causing a disease, or what specific gene locations are affecting your likelihood of acquiring cancer. And so I've been working on a general framework and algorithms to identify such relevant features. And the baseline of it is the extended random forest, which was something that we've published and I've since presented to the Mayo conference. It is an algorithm that uses a random forest, which is a pretty widely applied machine learning algorithm, and extracts variable importance measures from it, and determining the small subset of features in the data set that are relevant to a particular researcher's interests. So we can now algorithmically determine or estimate the uncertainty around which a particular feature in a data set, and by feature I mean a particular metabolite or a particular gene location, right, is affecting an individual. We anticipate that these interactions between different features in a human setting or in an animal setting can be very complicated. So the standard univariate tests and studentized t-tests for significance fail to capture these higher order interaction terms. So what we have been looking at is hypothesis generation through a combination of a maximal information criterion as well as the extended random forest procedure that we've already developed. And so the maximal information criterion is a technique we've implemented that can, in the same way that a Pearson correlation can attribute like an R-squared type value that assesses the linear correlation between two features, we can now assess different types of association. So as you can see that on a ranking between 1 and 0, we can now algorithmically assign a signal-to-noise ratio. We then present these as feature networks in a Fletcher-Reingold force-directed map to then suggest possible causes for diseases that we uncover in our data sets.
All right, time. Question? Henry?
So how is your model evaluating against other models? Does it achieve better performance?
How does your model evaluate against other models?
Right. So a traditional machine learning problem for supervised learning is trying to reduce generalization error. That is not the case for us. We are trying to uncover variable importance for the purpose of hypothesis generation. So they are different end goals. That being said, we have established some PAC bounds on the learnability of this algorithm. Although they're pretty wide bounds, we do have statistical significance in stating whether a feature is relevant to the hypothesis of it being included in a model.